Amino acid have an amino group, NH2, and carboxyl group, COOH, that are bonded together, okay, or to the alpha carbon. What is the alpha carbon? This is a good question. The alpha carbon, or even before going to the alpha carbon, okay, the alpha carbon is a symmetrical carbon that is found in an amino acid to which an amino group, a carboxyl group, and a side chain, which is R group, and even a hydrogen are covalently bonded. So this is very important to know it. Again, the alpha carbon is a symmetrical carbon in an amino acid to which an amino group, a carboxyl group, a side chain or the R group, and a hydrogen are covalently bonded. So this is the alpha carbon. So if we are talking about the amino acids, we can also say that amino acid in solution at a neutral pH, they are mainly dipolar. They are mainly dipolar ions. That is, they possess a positive charge at one end and a negative charge at the opposite end. This is the meaning of the dipolar ion. So amino acids in solution at neutral pH are considered or are mainly dipolar ions, which means they possess a positive charge at one end and a negative charge at the opposite end. So generally, or this is generally how amino acid exists at the cell or at cell pH. So each carboxyl group, COOH, donates a proton and become ionized, whereas the amino group becomes ionized as it accepts a proton and becomes a NH3, as you will see here. So each COOH donates a proton and becomes COO negative, and each NH2 accepts a proton and becomes NH3 positive. So if you are looking here for this diagram that illustrating the amino acid at pH 7 in a living cell or at any living cells, amino acids exist mainly in their ionized form as a dipolar ion. Why? Because of the ability of the amino acid carboxyl group to accept and to release proton. The amino acid in solution resist change in acidity and alkalinity and are therefore important biological buffers. Because of this, again, because of their ability or the ability of amino acid, okay, or the amino and carboxyl groups to accept and release protons, the amino acid in solution resist change in acidity and in the alkalinity and are therefore important biological buffers. So if we in detail talk about the different portions of the amino acid, here is the alpha carbon, and here there is amino group, and here we have the COOH or the carboxyl group, and here we have the hydrogen, and here we have R group. In this case it is the methyl or CH3. Okay, it can be anything or any R group. We have 20 amino acids that are found in most proteins, and the amino acids are grouped by the properties of their side chains. So they can be classified either non-polar side chains that are hydrophobic, water hating, or it may be polar side chains, and in this case they are hydrophilic. Hydrophilic means water loving. So a side chain with a carboxyl group is acidic, while the side chain that accepts a hydrogen ion is basic. We will discuss this now. So an acidic amino acid has a side chain that contains carboxyl group. Okay, so at cell pH, the carboxyl group we did mention before, it's dissociated, giving the R group a negative charge and A basic amino acid becomes positively charged when an amino group in its side chain accepts the hydrogen ion, while acidic and basic chains are ionic at the cell pH and therefore they are hydrophilic. Okay, so hydrophilic means water loving. That's why they are acidic, or because they are acidic, they are hydrophilic. Okay, if we are going to discuss the different forms and the different shapes of amino acids, for example, here, those are example of common amino acids. This group representing non-polar amino acids that have side chains that are relatively hydrophobic. We can even proceed illustrating this by also saying the standard three-letter that or and the one-letter abbreviations appear beside the amino acid indicating its name. For example, here we have the leucine. This is the name of it, and there are the first three letters of that representing the leucine as well, and even L is also abbreviation for leucine, which is an amino acid. This is the chemical structure, and this is the model that representing it. And if you are going even for the proline, which is a non-polar amino acid, there is here the three letters indicating its name and one letter also that referring to the proline. Here we have the valine or VAL as the first three letters, or only V that also representing or identifying or indicating the valine. Also we have the alanine, ALA, that also indicating alanine, or A that as well indicating the alanine. So if we proceed, this figure has the common amino acids as well, but here representing the polar amino acids that have relatively hydrophilic side chains. Relatively hydrophilic side chain. And a lot of lines under the word relatively. Because if we are looking here, okay, we will see that the carboxyl groups, and amino acid groups are electrically charged. Those are example of acidic that are here in the case of aspartic acid and glutamic acid. Those are acidic forms of amino acids. Also, we have basic forms of amino acids. Those basic forms are the methionine. Okay, no, not this. This is continuation. Here, the histidine. If you are looking here for this histidine, it is an example of the basic amino acid, and it's also a hydrophilic. So the acidic, the basic, and the polar are all hydrophilic or water-loving amino acids. Some proteins may have, or they already have, some unusual amino acids in addition to the 20 common ones. These are rare amino acids are produced by the modification of common amino acids after they have become part of or after they become a part of a protein. For example, after they have been incorporated into collagen, the amino acid lysine and proline may be converted to hydroxylysine and hydroxyproline. So this is a rare case. These amino acids can form cross links between the peptides or between the peptide chains that make up the collagen. So such cross links produce the firmness and great strength of the collagen molecule, which is a major component of cartilage, bone, and other connective tissue. So, as I mentioned, it is rare amino acids, and they are produced by modification of common amino acids after they have become part of a protein. As I just mentioned also, for example, after they cooperated or incorporated into collagen, lysine and proline will be converted into hydroxylysine and hydroxyproline. That makes the firmness and the strength of the collagen molecule. That is a major component of our cartilage, bone, and the rest of the connective tissue. Essential amino acids. They are those an animal cannot synthesize in amount sufficient to meet its need and must be obtained from the diet. Again, the essential amino acids are those an animal cannot synthesize in amount sufficient to meet its need and must obtain from the diet. So, animals differ in their biosynthetic capacities. So, what is an essential amino acid for one species may not be for another. So, differ according to different species. The 9 essential amino acids for humans are isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, and histidine. Those are the 9 essential amino acids. But arginine is added to the list for children, especially. Why? Because they do not synthesize enough to support their growth. So we can say now in the children there will be 10 essential amino acids.